We have 50 minutes or thereabouts, so we need to start pretty much on time. I have a few little words to say before we begin. Firstly, welcome all of you to the second Library Lunchtime lecture. I'm very happy to have Professor Tayfun Özçelik from the Department of Molecular Biology and Genetics speaking today. The title of his talk, Walking on All Fours, Evolution and Human Disease. It's a very exciting topic in general. Myself, I find genetics an extremely exciting and interesting topic, and I recently discovered that it has implications for studying the past. I am an historian, and I did not realize that you can study our own DNA to reconstruct movements of peoples hundreds or thousands of years ago. So it's something which I'm excited about, as I say. Indeed, the discovery of the structure of DNA by Crick and Watson in 1953, I think it was, in my opinion, is possibly the most important scientific discovery breakthrough of the 20th century. It has implications for all sorts of aspects of our lives. Professor Tayfun Özçelik has a medical doctorate, MD, from Istanbul University. He then studied in Munich before doing postdoctoral work at Yale and Stanford universities in the US. He returned then to Turkey, where he worked at Istanbul University, Department of Genetics, and he was chair of that department for a while, and he joined Bilkent in 1995. His research includes, and I shall read this list, but hopefully I pronounce things correctly, human molecular genetics, gene mapping, mutation analysis, identification of disease genes, and X chromosome inactivication. In addition, I should add, he has a very artistic side, and the image for the posters we've been distributing out here were in fact put together by Professor Özçelik himself, so he's a scientist and an artist at one time. Today's topic, evolution and human disease, he will focus particularly on a condition known as Unertan syndrome, and I won't say much about it now because he will explain it, but it has significance for Turkey, or significance for us I should say, in two ways. Firstly, it was first discovered in Turkey, and secondly, the results of his recent research with his team identifying the first mutation, genetic mutation, which caused this condition were published this month in the Proceedings of the National Academy of Sciences of the US. This is a copy of it here. You can find this through our library electronic resources. You can read it there. I will stop there. I will now remove myself bipedally on my 2 feet on the floor, and I shall let Professor Özçelik take over.
Thank you very much for this very kind introduction. I'm humbled. Actually, this is, I've never given a lecture in such a nice place surrounded by art, so this is an additional excitement. And also, I'm the second speaker in this series, and it is difficult speaking after Talat Alman. You know, I feel like Elizabeth Taylor's seventh husband now. I know what to do, I know how to do, but I don't know how to make it interesting. So, we are studying human genetic diseases throughout my career. I have studied, and in 2005, a most interesting family came up. This was discovered by Professor Ünertan, who is at Çukurova University in Adana, and since then we have been performing mapping studies and disease gene identification approaches to find the molecular basis of this interesting disease. To start with our story, actually, probably it is best to start with Charles Darwin. I want to take you about 150 years back, and at that time he made his voyages in the world, and after returning back, this is his handwriting, he wrote this historic document, The Origin of Species. And this is regarded as one of the most important scientific achievements in the past century, actually. And together with Mendel's work, who was in Czechoslovakia at that time, this laid the foundations of human genetics. Actually, the message that I would like to give you in brief is that when Darwin spoke about the origin of species, he actually never talked about humans, where they come from, and so on. But obviously, to mind, this had many implications, including these satirical depictions of him. This is the time of Victoria, England. Actually, at that time, they were really in competition with Germany in every field, including science. And at about the same time, in the Neanderthals Valley in Germany, at the limestone excavation sites, they discovered a skull. And this was regarded as a significant discovery immediately by the German scientists. And this is the Homo neanderthalis. Actually, we have a very close cousin, the Neanderthal man, which is extinct now. If you look into our history, you know how old is the world? It's about 4.6 billion years old. And when did first life arise on this planet? Probably after 1 billion years, after the creation. And then, the first bipedals, it was about 3 million years. Now, Neanderthals lived about 350,000 to 50,000 years. And after the second ice age, when there was a second flow of Homo sapiens from Africa, they came to Europe. And there was, prior to the second ice age, actually, I should add first, some of the early hominids moved from Africa to the world, including Europe. And then the ice age came, so they were separated. And the ones in Africa evolved as Homo sapiens, and the ones in Europe evolved as Homo neanderthalis. And then finally, they met about 350,000 years ago, and at the end, the Homo sapiens toppled down the Homo neanderthalis, and the Neanderthals became extinct. There is a lot of discussion whether there was inbreeding or not at that time. But there is now a Neanderthals genome project that's conducted in Germany to really answer all of these questions. The first phase was very exciting, but then they found out that it is actually the DNA of the technicians who were doing the sequence. Working with extinct DNA is very difficult. So they are working. But also today, nowadays, why Homo sapiens killed all the Homo neanderthalis? One, probably to relieve the guilt, I don't know, or maybe it's true, it's up to you. Homo neanderthalis probably never expressed himself through art. And Homo sapiens expressed himself through art. Now, anyhow, when Darwin wrote his piece and the Neanderthalis man was also discovered, the thinking minds immediately made the conclusion that humans actually evolved from apes. But then, of course, there is not an ape that comes and turns into a human overnight. So this is a long, long process, and there must be some intermediate species in between. And this would be, since there isn't any one now alive walking, then it is the missing link concept. And when this concept was introduced, based on the anatomical structure of the Neanderthalis man, a prediction was. And they said, well, if we compare the jaw and the brain, we should find such a fossil record that the jaw must be bigger than the human and the brain smaller than the human. Or vice versa, when you compare it to the apes, the brain should be bigger, the jaw should be smaller. So it was the brain that people thought is the enlargement of the brain is the key determinant of going on the way to Homo sapiens. And meanwhile, I put this is a, of course, important story. There was also one of the biggest scandals in history, science history. We recently lived one like this also, when the stem cell researchers' findings could not be duplicated. So these are magnitude 8, 9 earthquakes in science. Charles Dawson is a guy who was in England, and he provided the perfect missing link, perfectly fitting to the predictions, and the Royal Academy immediately recognized him. But until the 1950s, when the DNA was discovered, the Piltdown man was regarded as the missing link, despite the fact that researchers in other places were producing other types of missing links. They were always disregarded. Other prominent journals never published their findings, and this resulted in, of course, tragedies for those people who were working on it. But at the end, what came out is that this was a fabrication, the Piltdown man. It was perfectly fitting the predictions. They did carbon aging. In fact, when they first made the drillings, the flesh was even only a few thousand years old. But anyhow, to make it a long story short, what we learned throughout this time is that there isn't a single missing link actually. There are many different types of missing links from the early hominids, apes, to Homo sapiens today. And also, the hypothesis itself evolved. And in the first 90 years, it was regarded as the brain size. But the fossil evidence, by the way, the only way of studying such things is to look at the fossil evidence. The remaining from that time is the fossils. Today we have the DNA also, so there is molecular anthropology. But at that time, this was not possible. And findings did not support this. So, from 50s, this was when the Piltdown man scandal was revealed, until the 70s, the concept changed. And tool usage was regarded as the key evolutionary step for transition from ape to Homo sapiens. And therefore, all the excavations in Africa concentrated on the geographical layers where the tools were discovered to find the fossils with the tools. But later on, with the discovery of the famous Lucy, or Australopithecus afarensis, Lucy in the sky with diamonds was playing at that time, the concept changed, and it still remains that actually the first critical step is bipedal locomotion. Why? Because our brain development is also heavily dependent on the motor development. And by freeing the hands, fine motor skills start developing. You know, there's all these different mapping studies are done on the brain today, and which center is hearing center, where do you see? You see here, for example, occipital lobe. The motor cortex here is just here, as I'm showing you. The hand and the mimics and the tongue, the area is the largest, for example, when you compare it to the leg movements. And this, when the hands were removed from the could develop. And this triggered a set of events that also triggered the development of the mind, I would say. So this still holds that it's bipedal locomotion. Now, to study evolution, there are different types of approaches. The Human Genome Project and Model Organism Genome Projects, in the world today, we are sequencing the DNA of all the organisms. And the central goal is now to make, this is the NIH goal, to bring down the cost of sequencing to a dramatically low level that we can sequence anything. To give you a feeling, the Human Genome Project cost was $3 billion. In 2007, it became $1 million, from $3 billion to $1 million with the development of technology. And within the next 4 years or 5 years, the goal is to make it $1,000. And this is only possible with nanotechnology. Anyhow, so this is one approach. Another approach is the genetic diseases. And the concept here is genetic diseases affect the function of the genes. So, if we find diseases and identify the genes associated with those diseases, can we find key elements in development, in evolution? For example, brain enlargement. There are families, consanguineous mating through consanguineous, where there is a proportional microcephaly. So the enlargement of the brain, which gene triggered this? Speech is a unique characteristic we have. And there is a family, this is very famous, it's all out, Kearney family. It's also in our textbook that we study in the university. The Kearney family is not capable of speaking properly. They speak like they are drunk or disarticulate. But they have no brain abnormality, they have no mental deficiency, they just can't make the fine motor movements of the tongue and the vocal cords to express themselves in a proper manner. So, is there a speech? In fact, in 2001, in Nature, the identification of the first speech gene was published. It's a 4K domain gene. Nobody knows the function today. But also, when the evolutionary development of this gene was studied by analyzing the sequence of that gene in different species like the human, the chimp, and the gorilla, and so on, and mouse, they found out that actually FOXP2 only changed twice in history, and both of these changes occurred in the human lineage. And today, there is no one in the world who is not carrying such a change. So it is probably a very significant evolutionary pressure. Probably, maybe the Neanderthal man was not speaking, and during their fights, they started speaking, and communication was better in the fights. This is the Pakistani family where there is microcephaly, and a Harvard group studies this gene, and indeed they found another gene that's associated with this phenotype. Now, of course, quadrupedal gait is the key element in studying evolution and human genetic diseases. And this is how the disease was identified. This is the guy who identified it. He's a professor of neurophysiology, as I said, in Adana. And the reason that I'm showing you these hands is, although it's not really visible here, they are heavily calloused here, nasırlaşmış. And this shows that their quadrupedal locomotion is not a joke. That this is how, because everybody when they see such a thing, it becomes like a circus event, like from the Victorian times. Like when the hippopotamus came to Europe, it was a spectacle for everyone. And it was picked up by everyone in the world, including scientific journals like Science or National Geographic and BBC and Corriere della Sera, wherever you want it. And in Turkey also, it was picked up. Okay, I will show you the...
In a remote corner of Turkey, 5 unusual siblings come to the attention of the world. Victims of a mysterious condition, pariahs in their village, they now face international scrutiny. And all because of how they walk, on their feet and their hands. What could possibly have caused this?
Okay. Well, a picture is worth 1,000 words. That's why I showed this. This is a BBC and Nova produced this in PBS in America. It was supported by the Howard Hughes Medical Institute and Google. And, actually, bipedal locomotion is unique to our species. And although there can be, for example, congenital abnormalities, this is the dog face who brings traffic to a standstill, walks bipedally, no one, except for humans, are walking bipedal. There is more important, you know, we already know that it is our cerebellum here. When the patterns of inheritance wasn't known, if you cut the cerebellum, inside looks like the tree of life. And everybody thought that cerebellum is where all the genetic characteristics are Vorvita in the early days. In fact, today we all know that cerebellum is very important for our balance. And there are many disorders with cerebellar abnormalities. The thing is, they discovered, the Italian neurologists and the German neurologists recently discovered patients who have no cerebellum at all. So it is possible to live without a cerebellum. But these people, they walk bipedally, not quadrupedal. They don't have a cerebellum, they don't have such an intrinsic feature of our species to walk upright. Well, what are the clinical findings of this disorder? They are walking on all fours. And by the way, I want to tell you, this is a very efficient, it is performed in a very efficient manner by these people. Later I will show you one more movie. Another family was discovered in Iraq. They can go from Baghdad to Kerbela, this is 60 kilometers, quadrupedally. So they can do both the lateral and direct walking in a very efficient manner. Of course, they are profoundly retarded mentally. The brain is somehow smaller. In the cerebellum, there is a particular portion, the vermis, it's in the middle, which is smaller. And dysarthric speech, like a drunk person. But they don't have any conscious experience. I can tell, like, you say, what is your name? And they say, Mama. And you say, what day today? They say, 2. Or, they can use a few words, but they don't give you a logical answer. So they have a limited type of vocabulary. By the way, another important feature, they don't have any autistic features. Autistic features, I mean, when you approach to different types of neurological diseases, people affected with these diseases, they behave like autists, they don't want you. These are not like that. They are very curious, courteous, and also they come next to you and they investigate you. So nice people, very nice people. Well, when this was discovered, of course, we geneticists always first look at the family tree. This is the first step to go ahead, also for my students. Any type of significant paper that I published, it was because I studied the pedigree very carefully. I always based the first starting step on the families. So always go to the basic, look at the families very well. Now, by looking at this family, for example, this is, we call one family, but there are actually other composed of 5 different families. The inheritance pattern is picture perfect recessive inheritance, meaning the mother and the father should be carriers, and that there is a 25% risk of producing an offspring, a child with the condition. So this means that it is possible to go after this gene. So we initiated a genetic mapping study here. But as I said, since the condition was very heavily covered in the press everywhere, so everybody had interest in this family. And we had a competition with the German Genome Center in Berlin and with Cambridge University and London School of Economics, because London School of Economics has a Diamond Institute. And in December 2005, when we went to Adana, this is in Adana, and brought the family's DNA samples here and mapped the gene, the next day we learned that actually the Germans were there before us and that their paper is already accepted in Journal of Medical Genetics. So this was a big setback for us. You know, there are 2 steps. First is to map the chromosome and then find the gene. This is now only the first part, identifying the chromosome that is associated with this disease. And you know, nowadays, I want to also give you one more information. We are already using nanotechnology, and such mapping studies took, in the up until 2000, it took about 10 years, 7 years or so. For example, the Kieran family in London, they used the highest, the best available technology, and they were able to map the disease in 7 or 8 years. Now, with the microarray analysis, which is looking simultaneously at 500,000 points in the human genome, you can do such a mapping study in a week. So, from 7-10 years to 1 week is big improvement. And indeed, we also mapped it in 1 week. This was like, I want to make this analogy now. This is controversial, I know, but actually the DNA of these families also left our country without any ethical approvals or material transfer agreements. This is in fact a general problem now. Since consanguineous marriage rates are high in our country, identification of disease-related genes, we have a perfect environment for this. And it's a very, I don't here promote consanguineous marriages at all, but this is already the case. And finding, after the Human Genome Project, it is a major goal of the scientific community throughout the world to identify what a gene does. And you understand what a gene does by finding its mutant forms, its faulty forms. And this is through diseases. So there are about 5,000 single gene disorders in the human genome, and among those, only about 2,000 are mapped today with the gene identified. There is still 3,000 different diseases. This is a major issue in medical genetics impacted medicine. And this is like making the Pergamon Museum again in Germany by giving the DNA samples. Anyhow, then something magical happen that any geneticist dreams of. This is to find more families afflicted with the disorder. Here, I should mention that press was critically important because in any type of genetic disease, what you identify is always the tip of the iceberg. And there are many people who are affected with this condition who stay at home. But when the press starts covering, they come out and they say, oh, we know someone with quadriplegia and so on. So in Turkey, this was the first family in Hatay, and then in Adana, in Gaziantep, and in Çanakkale, further families were discovered. And we resumed gene mapping, and as I mentioned, we used this gene chip approach for all of these mapping studies. And what we found was very interesting. Why is it very interesting? Let me tell. For example, let's move hearing impairment, sağırlık. There are sağırlık, it is a genetic disease, but there isn't a single gene for hearing impairment in the human genome. There are actually 28 different loci that are known. There is a gene on chromosome 7. When it is mutated, people become deaf. There is another gene on chromosome 9, and in some patients, it is the chromosome 9 gene defect. So we call this concept the genetic heterogeneity. Different disease, different genes, mutations of these genes can cause similar phenotypes. In fact, this was the case also for quadriplegia, and that in different families, different chromosomes were implicated. This is we call homozygosity mapping. Homozygosity meaning we compare the chromosomes of the affected siblings with the carrier parents and also normal siblings, and we look at those regions of the chromosomes where the affected ones inherited the same chromosome from their parents, homozygosity mapping. And chromosomes 9, 17, and 13 was identified. This is the same chromosome that the Germans also identified in one of the families. So when we looked into the chromosome 9 region, we found an obvious candidate, which can be now further studied. This is now, I'm telling you the gene that we discovered. It is the VLDLR gene, the very low density lipoprotein receptor. This is actually a very famous gene. It is in the cholesterol metabolism. And there is a specific type of mutation, and the mutations are also different varieties of mutations. This was the perfect type of mutation because it caused a stop codon, what we call as stop codon, directly stopping the synthesis of the protein. A perfect, no doubt mutation. Some mutations are missense mutations, although their effects are very deleterious, you just don't know without functional studies. Well, actually, of course, finding a mutation alone is not enough. One has to study people, normal individuals from the same geographical area. So we studied like 500 different people who came from Gaziantep and also from Çanakkale region. And we found that in the general population, this mutation doesn't exist. And we've done some functional studies, like we asked what happens when this mutation is present. Does the protein get synthesized? The protein is on the membrane, does it go to the membrane? And we found some answers to these questions as well. So, it is indeed the very low density lipoprotein receptor. So I want to take a closer look. Actually, cholesterol metabolism is the most decorated pathway in medicine, have been awarded to studies of cholesterol. This is the first one in 1927, 1928. These are actually the chemistry prizes. and then in medicine 1964, and in 1985 the Brown and Goldstein's discoveries. In fact, these are researchers that are at Dallas, Texas, Southwestern Medical School, where many of our students go for their PhD studies, and some of them are their students now. And they are the ones who actually discovered the VLDLR gene. In fact, today, many of the cholesterol-lowering drugs that many of us use, the target molecules were identified by these two receptors so that medicines could be produced to lower the cholesterol. In fact, Brown and Goldstein had published a paper in 1995 where they produced a mutant form of this gene in the mouse. This is also another basic approach in biomedical sciences. When you have a gene in your hand, you introduce a mutation in the mouse, a mouse model, and you look what happens. And they were expecting to have profound implications in the metabolism, meaning their lipid levels should be different, these mice. They probably have heart attacks very early on. They should probably get obese, who knows. But what they found is there is no phenotype. And their lipid metabolism is normal. And it was an interesting situation. Normal plasma lipoproteins and fertility in gene-targeted mice homozygous for a disruption in the gene encoding the very low density lipoprotein receptor. This remained a mystery until 2000 or so, when the same group, Brown and Goldstein, made another discovery. You see, when a gene is doing something somewhere, that's what you think. Its real function could be totally different. And lipoprotein receptors also have such a characteristic that they function in the nervous system. For our neurons, when the brain is forming, the neurons migrate in the brain to specific locations. And this is through chemoattractant signals. These chemical signals of the stem cells, they call the developing neurons, come here, huh? And then there is a lot of apoptosis. Apoptosis in autumn, all the leaves fall down, for example. It's a programmed cell death. So many neurons are also dying at the same time, so the architecture of the brain emerges. Now, there is a central protein, reelin. Reelin is such a message in the brain that it's synthesized by a gene, it's a protein, and reelin does its function by binding to the very low density lipoprotein receptor and then triggering all the cascade of events. So, actually, through this study, we had the first identification of a gene that is associated with the bipedal locomotor abilities of humans. So this is a genetic disorder. But the controversy that exists in science also existed in this study. And that some people are making really, to us, peculiar comments. For example, to put this is written by them. This is Nicholas Humphrey in London School of Economics, a prominent scholar in evolutionary studies. He thinks that quadrupedal walking in these people have emerged as an environmental influence on these people. And what could be the environmental influence? Well, the environmental influence is, since these people are in Turkey, and since Turkey is predominantly a Muslim country, and since there is a teaching of tevekkül or acceptance, when these children are born, their parents say, this came to us from Allah, we have to do nothing, we should do nothing about it. They accept the situation, and these children become quadrupedal. And he also says, you see, we discovered
The
another...
story, unlike any other you will ever hear from Iran, it's not about death or war or destruction. It's about 4 siblings from a village near Eilat, south of Baghdad, who have never learned to walk on 2 feet. They crawl a bear-like crawl on all 4 limbs. There is only one other family we know of in the world who does this, and they're in Turkey. We couldn't visit them in here, it's far too dangerous, so they came up to Baghdad to see us. And you can see them here behind me. There is Hussein on the right. He can walk a little bit. He can walk the best of all of them. There is Ali and Abbas, who can stand up, but they can hardly walk. But they are magnificent on all 4s. They recently walked 60 kilometers on a holy pilgrimage to Kerala. They're a bit backward, their speech is slurred. When they were young, they were teased by all the other boys in their village, but they fought back and they've earned their respect, and they're now accepted. They can't read, they haven't been to school, but they're lovely, lovely men who joke and laugh with each other. Their father, has done his best to get medical help for them. He's got all the doctors, the best doctors in it.
What they do say is actually also when Zimmer frame is given. Zimmer frame is, you know, the thing that you hold in front of wall. They become bipedal, and nobody gives them the Zimmer frame, so they are quadrupedal. But I mean, this is really absurd because if, for example, glasses correct my vision, then is my myopia environmentally induced? For example. And when in the presence of a gene, plus now there are in Turkey, in Iran, in Brazil, in Iraq, in Mexico, in India, these are being reported as a genetic disease, also solely on the geographical distribution, it's a genetic disease. But of course, such things make publication of a paper really difficult. And when we sent the paper to Nature first, it went out for review. But the reviewers, some of them were really negative because of competition and so on. But thanks to Mary Claire King, who is a prominent member of the American Academy of Sciences, who is actually the discoverer of the breast cancer gene, actually, and other people who were in the editorial board, like Uta Franke, and Goldstein himself, who first discovered and got the Nobel Prize for the cholesterol metabolism. We got our paper accepted in PNAS. So, in the last 5 minutes, I will go, what next? Since now we have the paper out, what is the next step? Other genes, other diseases, genome evaluation, and medical implications like career identification, prenatal diagnosis, functional studies, and molecular targeting for treatment. These are the things that can come to my mind immediately that can be done. One is, I want to tell you a direct example. When the FOXP2 gene was discovered in the London family that has a speech disorder, actually we have this concept in genetics, hypomorphic alleles, which means that the same gene can be affected with different mutations in different diseases. For example, dyslexia. Some of the dyslexia patients actually have this mutation of the same gene. So, is it possible that DLDLR, the very low density lipoprotein receptor, has other diseases? Like, for example, in the Hutterites, this is people who migrated to the United States from Austria. They have a specific disease with a chromosomal abnormality, gross abnormality that involves the DLDLR gene. This is the renowned physician from Sweden, Bent Hackberg, and in the about 25 years ago, he described a disease called disequilibrium syndrome. These people have equilibrium abnormalities. Maybe the same gene, nobody knows the answer today, but maybe hypomorphic alleles of the same gene could be affected in disequilibrium syndrome. Another feature, now we can look directly at the molecular evolution of our gene, just like they did for FOXP2, comparing the sequence of the gene in different organisms. We are comparing the sequence of the gene and looking at the specific changes that alter the amino acid composition of the proteins. This gives us information on whether evolutionary pressure exists on a gene or it doesn't exist. Such type of anthropological studies at a molecular level are possible. Here. This is already, this family is part of the teaching curriculum in England and in America. There is a website designed for this, and also how to teach it, how to organize a course, the activities, and what kind of neurological tests, and also interactions among family members, what they believe in, and how to organize the students to study this family. Now, of course, this is a beginning for us. We have two more genes. One is on chromosome 17 and another one on chromosome 13. So I think identification of these genes will be also critical in identifying the molecular pathways that are involved in the development of the cerebellum. At present, our understanding of the cellular events in this important organ is just the surface we know, and through these families we will probably find these in a more detailed way. Last, I would like to mention to you who took part in this study, this is very important because actually our students were competing with the German students. And it really needs a lot of hard work. And here, Elif did the mapping studies of the sort of. Actually, they were in every part, but predominantly. Shafak is here. Okay.
We would like to see them.
Yes. Okay. Elif, let's start. And Shafak did the identification of. Süleyman is there. He's our computer guy. You know, you need a lot of bioinformatics. For example, you generate how many gigabytes of data by one genome screening? 5 gigabytes. 5 to 7 gigabytes of data is produced. So analysis is important. And Emre did the and of course in the lab we have Çiğdem and Melda. Where are they? I have to mention one more colleague. These are always collaborative studies. Nurten Akarsu here. She's a professor at Hacettepe University who was critical in our mapping studies. And also, actually this time, it always happens like that, all the samples, drawing the samples, my wife gets fainted when she sees it, but this time it didn't happen, and she was with me all the way in Adana and Gaziantep, and we collected all these samples together. This is the story so far, and we hope in the future we will have more genes identified and more to tell. Thank you very much.
I'd like to thank Professor Özçelik for what was an extremely interesting and very comprehensible talk, where most of us are not specialists in this subject, and I followed all of his talk. I was very excited to know about that. Our time is pretty much up, but I'm sure if you have any questions individually, you can speak when we've finished. We have to finish at 1:30 today. We'll stay and have a brief discussion with you if you have any particular questions for us. So we won't have any public questioning now because people have got classes to get to, I realise. But once again, thank you to Professor Özçelik and his team for telling us about this exciting genetic detective story as it is, and we wish you the best to crack the code and finish the story in the next few years. Okay, thank you again. Our third and final library lunchtime lecture for this semester will occur in 3 weeks today. Professor Ekmel Özbay will tell us about nanotechnology and why it is so hot. And in fact, nanotechnology was mentioned a few times today. I'm very excited to hear about that.